Okay, so welcome back to返學 DSE, and we are now in chapter 14 talking about growth and development. And these two words actually have different meanings. So first, let's talk about growth. Growth is an irreversible increase in size and dry mass, and it can be resulted from two different events. One is the increase in the cell number, another one is the increase in size, that is the cells getting bigger and bigger, or we have more cells. As for development, it is brought by differentiation, that is the increase in complexity. Let's say the cell in the brain and also the cell in the lung, they have different special functions, so we call that differentiation, and this term is also used in mathematics, as some students may also take M1 or M2, and the cell becomes specialized to perform a particular function. That's what we use differentiation in biology. So let's talk about plants first. So for plants, the most obvious group is from a seed. So for seed, we have seed coat, mentolus, and also the hymnus. For these terms, they are no longer that required in your curriculum. So if you can remember it, that's great. If you can't, just leave it. For these terms, you have to remember it better. That is for the pilus and also radial and also the cotyledon, because for these three terms, they are combined as known as embryo. And for the cumulus, that is developed into the shoot, and also the radicle will be developed into the roots. As for the cotyledon, it contains the stored food for growth. So from a seed to a plant, this process is we call the seed germination. This process is actually requires some condition. So what are the conditions? Number one will be water. It is used to dissolve the seed coat and also make the cotyledon swollen, and this will activate the enzyme in the seed, and also the water will act as a reactant for the hydrolysis of insoluble foods such as starch, and also the water will serve as a medium of transport of soluble food to growing region. And the second element required will be the warmth, that is a reasonable temperature, because the enzyme need to work effectively based on the temperature. And third one will be oxygen, because all organisms will require respiration, not surprisingly, including the seed, and respiration will provide the energy necessary for growth. So here are the stages of seed germination. First, the seed coat will break and the radicals emerge, and then the cotyledon will turn green for photosynthesis, and then as the first leaf actually is generated, and then the cotyledon may fall off. So in order to result in growth and also development, the plants will have the meristem. This is a group of undifferentiated cells, and they are here to divide by mitotic cell division throughout their life. Because for mitotic cell division, all the seed daughter cells will be identical as the mother, which are also undifferentiated. So as the cell migrated, they will become differentiated. So there will be two types of meristem. One is the apical one, another one is the lateral one. So let's use this figure to illustrate. For the orange arrows, it is the apical meristem. It results in an increase in length, and we regard it as the primary growth. As for the blue arrows, it is the lateral meristem. It is the increase in thickness, and we regard it as the secondary growth. So let's focus on the root first. So in the root from top to down, that is the very tip of the root, we have the region of differentiation, region of elongation, and also region of cell division. As for the cell division, it contains the meristem, so it continues to produce the new cell by mitotic cell division. And because the cells are very condensed, so it doesn't even have a vacuum. And then it will migrate back, and then it will become the region of elongation, because as the water enters by osmosis, and then the cell will be elongated and stretched, and it will push the apical meristem downward. And then we will come to the region of differentiation, because in this region, it will start to appear the vascular bundles and also the root hairs. So at this moment, the cell will become differentiated. That's why it is defined as the region of differentiation. Similarly, there will be also three regions can be found in the shoot. However, for the peripheral of the stem, that is the lateral one, lateral mirror stem. The secondary growth is governed by the vascular cambium, and it is commonly found in the woody plants, that is the trees that we see. So here is a labeled diagram, and the vascular cambium is labeled as F, that is this region. And for the D and E, they are xylem and also phylum respectively, and you learn it in chapter 10. As for A, that is the epidermis. As for the B and C, they will be the cortex and also the pitch respectively. So you have to understand the labels in this diagram. So I use this cartoon to illustrate the idea of secondary xylem and also secondary phylum. So in the cambrium, the secondary xylem will push inward and become wood, while the secondary phylum will push outward and become the phylum. And this results in an increase in thickness and also this kind of ring structures. And we can use this ring to, because for 1 year there will be one ring. And in order to measure growth, there are a few ways that is also known as the parameters. That is size, fresh mass, and also dry mass. As for size, we are measuring either length or height, surface area of the flattened structure, or volume. And the advantage of using size is that the object will remain alive and it is easy and convenient to do it. However, the disadvantage is that the growth in other dimensions may not be taken into account. Let's say you are measuring the length and height, then you may miss out the increase in volume. Let's say you are measuring the volume, then you may miss out the surface area increased. So here are some bias of the size. So more commonly, we will use fresh mass. That is, we will measure the total mass, including the water content. And the advantage is that, again, the object will remain alive, and also it is easy and convenient to measure the fresh mass. However, the disadvantage is that it is easily affected by the water content, which can vary with environmental conditions and also activities of the organisms. So in order to prevent such variation, we can use dry mass. That is the mass of the organic matters only. So we put the object into the oven and then we wait until there is no further change in mass, and then we will obtain the dry mass. The advantage is it is very accurate. However, by doing so, you will kill the sample. That's why you need a large sample size. Let's say you need 100 of the plants and then you kill 10 of them every single time. It is also very time consuming to wait for the dry mass. And because you have to put the whole object into the oven and wait for it to dry up, so it is not suitable for large organisms. So here is a growth curve of an annual plant, that is, its life is within a year. So it is somewhat like an S-shaped curve. So there will be 3 stages: the initial drop and then increase and then with a drop again. It is a competition between the consumption and also production, because initially you will use up the stored food, and that's why the dry mass will decrease. And then as the leaves is generated and the plants will undergo photosynthesis, there is an increase in the dry mass. However, as it approaches its end, it will store the energy into their reproductive organs, and also their leaf will fall off. That's why the dry mass will drop at the end of the day. And here is a growth curve of a human. And the y-axis shown in here is the height, and then x-axis shown here is the age. And we can roughly divide it into 5 different stages. The number 1 will be the infant, and then the second one is the childhood, third one is the adolescent, fourth one is the adulthood, and the last one will be old age. You may notice that in the infant and also the adolescent, the growth rate is very rapid. There is a rapid increase of the height. While in the second stage, the growth rate is relatively slower. And then in the fourth stage, that is the adulthood, your growth of height is relatively constant. And finally, in the old age, there will be a decrease in height. So it's also commonly found in our communities. So this was about a human throughout their lifetime. So let's talk about different gender or sexes. So we are comparing the boys and girls, and their increase in height annually. So you may notice that for girls, there is an increase of the height much earlier than the boy, because the girls will enter puberty earlier than the boys. However, you may also notice that for the boys, the peak is slightly higher than the girl. That's why generally the boys are a little bit taller than the girls. So don't be so racist or femalist about this biological function. And let's talk about the growth rate of different body parts. For the head, it grows rapidly in the early age. It enables us to learn, and relatively the size will decrease with age. As for the limbs, there's our arms and legs. It will grow relatively slowly in the early age, and the relative size increase with age. As for the trunk, there's our body trunk. It will grow at a rate proportional to the whole body, so the relative size remains nearly unchanged throughout our lifetime. So here is an extract of the HAEA document. In the topic of growth and development, we have to understand the difference between growth and also development. For growth is increase in number and also mass, while for the development is the differentiation. And then for plants, we learn about germination, and also we can use growth curve to describe the growth of both annual plants and also human. And in order to measure growth, we have a few methods. Let's say height, weight, and also fresh mass and dry mass. So here will be the end of the revision videos of chapter 14. I hope to see you guys next time. Bye bye.